Hello, Bio 104 students. In this lecture, lecture 6, we're going to talk about the origin and early evolution of life. Here's a vocabulary that you'll be engaging with for this lecture. Okay, so let's ask what is life, and more specifically, what is alive? These are difficult questions because we only know about life on Earth. And since we've already come to the conclusion that all life has a single origin, life is an ontological individual. So there's the possibility from the philosophical perspective that it cannot be defined. Now, we can approach the question perhaps simply by noting the key qualities of life as we know and understand it. So living organisms have the following properties. They have the capacity of reproduction. They have the capacity to evolve. They have a membrane or a coat that provides a barrier to the outer world. And they have the capacity to capture and transform physical energy sources into biochemical forms, acquire biochemical energy sources through predation, and the capacity to use biochemical energy to maintain and build the organism. That is negative, for example, negative entropy. Then we'll close the lecture by asking the question, are viruses alive? So how do you get from life to non-life? Well, it's clear that life originated from inorganic matter by non-biological processes. But what are these processes? And a key mechanism for these processes must have been self-assembly. And self-assembly is a process by which a system's components, molecules, polymers, are organized into ordered and functional structures as a consequence of specific local interactions among the components themselves without external direction. So an interesting example is a mechanism of self-assembly is phase separation. So think about the unmixing of an unstable chemical mixture, such as bubbles in beer, or oil in water, or vinegar and oil in the Yale dining halls, where they give you the salad dressing in a bottle that has an opening, and yet the phases is separated. There's no way to actually shake it to get your tasty salad dressing. Okay, let's look at geologic time. Now, this is a figure that we looked at in the short abbreviated lecture on the fossil record, where the point is that giving the 4.5 billion years of the history of Earth, a fraction of that, about 540 or so, have complex organisms in the fossil record. So what's happening during all this time prior to the appearance of complex organisms in the fossil records? Well, a lot was going on. So virtually all the fundamental attributes of life evolved in this Precambrian period. Although nothing was very big to where it was preserved in the fossil record, and we actually have a very poor fossil record because of nothing very big, and then another mechanism that we'll talk about after this slide. So here what we're looking at on the bottom, we're looking at the geologic time scale. So if we look at the Phanerozoic period here, you can see it's a small fraction of time. We're going to the middle part of the diagram now. And we're looking at the entire history of Earth, dating back to about 4.5 billion years ago. And we could see that all of the key aspects of the origin and evolution of life happened prior to the Phanerozoic. The origin of life itself, the origin of first cells, the origin of photosynthesis, the first colonial cyanobacteria, which invented photosynthesis, the first eukaryotes, the first photosynthetic eukaryotes, which basically boosted it from the cyanobacteria, the first multicellular eukaryotes, and the first fossils of multicellular animals appear prior to the Phanerozoic, all of this in this time period known as the Precambrian. So prior to the presence of complex organisms in the fossil record, starting at about 540 million years ago, many of the key aspects of the origin and evolution of life had already happened. Now, One problem is in terms of making it difficult to find old fossils is that it's really hard to find old rocks on Earth because of this process of the rock cycle, where you have magma, uplift, erosion, weathering, sedimentation, metamorphosis, plate tectonics, repeat. So this constant cycle, recycling of rocks on the Earth means that there will be very, very few rocks that date all the way back to the time in the Precambrian when all of these key events in the origin and evolution of life were happening. Now, the oldest sedimentary rocks are found in Greenland, and they date to about 3.8 billion years ago. And there are chemical traces in these rocks, even though it's disputed, that suggest life already existed. Now, this is controversial because it's hard to distinguish a fossil cell from other self-assembled inorganic spheres. In fact, the same problem of separating a fossil from other self-assembled inorganic spheres applied to the issue of the Martian fossils, which were noted and discovered in a meteorite, a Martian meteorite that landed on Earth. And I included the Wikipedia link to this controversy of Martian fossils. By the way, these were not, the Martian fossils were not deemed to be fossils, but it was in the scientific and popular dialogue, I believe in the mid-late 1990s. Now, there's interesting fossil evidence for early life, that early life, Earth's oldest life, occurred in deep sea hydrothermal vents. And this is taken from a paper published 3 years ago in Nature, and basically saying that these submarine hydrothermal vents, about 3.8 billion years ago, and possibly dating to 4.3 billion years ago. So based on chemical and morphological lines, they're arguing that these are fossils that represent the oldest recognized life forms on Earth. So if indeed these are the oldest life forms preserved in the fossil record, it would indicate that the origin of life was somehow associated with oceanic hydrothermal vents. However, there's actually stronger direct fossil evidence for the earliest life. So over here on your left, you have early cellular remains from South Africa that date to about 3.5 billion years ago. In the upper right, we have fossils dating to about 3.26 billion years ago of dividing cells. And then 3.5 billion years ago, fossils from Western Australia that are very similar to filamentous cyanobacteria. So these are examples of the oldest evidence in the fossil record for the earliest life. And they all date to a time period in the Precambrian of about 3.5 or 3.3 billion years ago. Now, we could think about the origin of life. If we wanted to construct an organism, we have these challenges. One, we have to assemble simple molecules into building blocks for complex polymers. We have to assemble the polymers so that they can store information and catalyze reactions. So we need information storage and biochemical work. Then we should add a membrane and an energy source to complete the makeup of a living organism. Now, very early hypotheses about the origin of life developed right after the publication of Darwin's book. So the premise that all life on Earth shares common ancestry and that there's a single origin of life on Earth, naturally people would then begin speculating about what were the conditions that led to the origin of life on Earth. So on your left here, we have a letter from Charles Darwin to his colleague Hooker, where he says life may have began in, quote, warm little pond with all sorts of ammonia, phosphoric salt, lights, heat, electricity, present so that a protein compound was chemically formed, ready to undergo still more complex changes. And then later ideas in the mid-1920s, people were talking about primeval soups of oxygen molecules could be created in an oxygenless atmosphere through the action of sunlight. In fact, prior to life on Earth, there was no atmospheric oxygen. And JBS Haldane, who we've talked about a couple of times in the course, basically thought Earth's prebiotic oceans were different from their modern counterparts, would have formed a, quote, hot dilute soup from which organic compounds could have formed. Now, these ideas came together in the early 1950s with the really famous Miller-Urey experiment. Now, Stanley Miller was a graduate student of Urey, who was a very famous biochemist, and they were working at the University of Chicago. And in fact, this work won them a Nobel Prize. So what they were able to do was synthesize prebiotic molecules in an experimental atmosphere. Now, let me show you how this experiment was set up. So this is a diagram of the apparatus that was set up in the lab. So in this flask, they combined water, ammonia, hydrogen, and methane. And in this flask, they exposed the chemicals in this flask, in the vapor, to electrical sparks that was to simulate lightning on the prebiotic Earth. And the chemical compounds that were created in this flask were then distilled and condensed, and they were basically collected and analyzed. The compounds were analyzed in the component liquid. I should have said prior, this was the oceanic compartment where the liquid was boiled to form the vapor that in this flask then was exposed to these electrical sparks. So what did they find? So when they analyzed the products of this reaction, they found essentially a fair number of amino acids and nucleic bases. So from these simple compounds, they were able to synthesize amino acids and nucleic acid bases and sugars simply from generating the vapor and exposing it to the electrical spark. They were able to make these complex organic compounds. Now, the Murchison meteorite fell on Australia in 1969, and pieces of these meteorite were dissolved in water, and the soluble components of the meteorite included amino acids, nucleotide bases, and sugars. So it's clear that these organic compounds could also have come to Earth from a non-Earth source. So all life on Earth is based on L-isomer amino acids, but the Murchison meteorite included a mixture of L and D isomer amino acids. Now this showed that these amino acids did not given the L and D mix, they were not contaminants from life on Earth. And in fact, if you look on the right, this is a comparison of the amino acids produced in the Miller-Urey experiment, here the discharge experiment, and those found in the Murchison meteorite. Okay, so if we think about the origin of life, we have been able to assemble simple molecules, and we can even form complex polymers. But how do we get to the second stage where we're assembling polymers that can store information and catalyze reactions? That is, molecules that can store information and do biochemical work. Now, what we need are life requires chemical catalysis like proteins and genetic information like DNA. So is there one molecule that can do both? That is, convey genetic information as well as do biochemical work that we see here on the right. And yes, the answer is RNA. RNA can convey genetic information, and we know that RNA can also do biochemical work. So this leads to a question: was the precursor to life an RNA world? An RNA world is this hypothesis that the primordial form, the early form of life, was RNA-based living system that later gave rise to DNA-based living systems. And so in our diagrammatic phylogeny, what we're showing here is that all of the lineages that are around today are in blue, And we're going to introduce Ida and Luca later in this lecture. That's what we would call Ida being the idealized initial Darwinian ancestor, Luca being the last universal common ancestor. So in the RNA world, RNA may have been the molecule that was doing information storage and information transmission, as well as biochemical work. The RNA world may have been the precursor to the modern world of the central dogma that we're all familiar with, where we go from DNA, RNA to protein. So given that RNA is the intermediary between DNA and protein, and RNA can indeed do biochemical work, this leads to the credible hypothesis that early life was based on an entirely RNA world. And what really led to the development of this hypothesis was the discovery of ribozymes, in part by Sid Altman here at Yale University, won a Nobel Prize for this work, in demonstrating that RNA can act both as a biological catalyst, like a protein enzyme, as well as carry genetic material. So we know that RNA, through ribozymes, can do biochemical work, and obviously through the role of RNA in the central dogma, it can convey genetic material, genetic information. So how did we get to an RNA world? So first, ribose, nucleic acid, and phosphates must have joined to form the first ribonucleotides. And these would have had to have undergone a spontaneous polymerization, a form of self-assembly. Remember, self-assembly is key to the origin of life. And it's hypothesized that inorganic catalysts may have been essential for self-assembly of the first RNA strands. And people have speculated that perhaps clay minerals were the inorganic catalyst that allowed this self-assembly of early RNA. Okay, now we have to think about adding a membrane and an energy source in the final steps of making our living organism. And this gets us to think about the origin of the cell membrane, which is critical to life. Early days in the 1960s, Sidney Fox in his lab was able to create proteinaceous protocells, which he called microspheres. And amphiphilic fatty acids have a polar head and nonpolar tails. In solution water, the fatty acid tails will avoid the polar water and the heads will seek the water, all right? So we make this, basically this bilayer. So fatty acids can self-assemble into a spheric bilayer with water inside. And more recently, others, including this lab at Harvard, have shown self-assembly or spontaneous formation of stable-like cell vesicles with permeable membranes that are capable of cell division, or capable of subdivision, pardon me. Okay, now if we look at the history of life on Earth, we're looking at a phylogeny of life on Earth here. These are the events in the Phanerozoic, that is, remember, the part of the fossil record that can, or the part of the geologic record that contains fossils. We go back to about 3.5 billion years ago. Based on what we know from these early fossils, there would have been abundant life about 3.5 billion years ago, and this life was DNA based. However, going further back, we don't know what life looked like. Perhaps it was this RNA world. Now, in thinking about the phylogeny of life on Earth, the most recent common ancestor to all extant organisms, we're going to call this person Luca, or this thing Luca, for the universal, the last universal common ancestor, okay? And the primordial form, that is the first living thing, we're going to call Ida. This is a typo here. I will correct it in the slides that are when they're posted. This is the initial Darwinian ancestor. The primordial form is the common ancestor to all life on Earth, whereas Luca is the common ancestor to all of the existing lineages of life on Earth. So by definition, Luca is younger than Ida. because we're presuming that there were other extinct lineages that are not Eubacter, Archaea, or eukaryotes. So going back to Ida, life was undergoing evolution by natural selection, and by the time we get to Luca, we have cells, DNA, and proteins. Now, another way to think about studying the origin of life is through phylogenetics. That is, by understanding the architecture of the tree of life, we have some insight into the origin of life. Now, here's a problem. Remember we talked about phylogenetics, we talked about rooting the phylogeny using outgroups. That is, we use lineages that are closely related to the lineage that we're interested in examining the phylogeny to root the tree. Well, the problem is pretty self-evident. If we're trying to do a phylogeny of all life on Earth, that is where Luca is the last universal common ancestor, how do we root that tree? We don't have outgroups. There's no non-Earth life that we think is related to life on Earth that would make Luca monophyletic that we could use as an outgroup. Now, we're going to review this a little more later, but just let me point out that the major clades in the tree of life include the bacteria, the Eubacter, include the eukaryotes, that's us, eukaryotic organisms, and the archaea, okay? The archaea, eukarya, bacteria. Bacteria and archaea are thought to be prokaryotes because they don't have the traits that are eukaryotes. That is, prokaryotes are not a monophyletic group. But back to the question, how do we root the tree of life? Well, we root the tree of life through duplicated genes. Throughout the history of life, there are gene families where those gene lineages duplicated prior to the speciation of the lineages. So for example, in your retina, you have different opsin proteins. You have a rhodopsin that absorbs light at low light levels. You have different colored opsins that are detecting light at different frequencies of the visible light spectrum. These different opsin genes, if you do a phylogeny of all the gene families, opsin genes form a monophyletic group, and within opsin genes, you have different monophyletic groups that correspond to all of the rhodopsins that we see in vertebrates, all of the green opsins we see in vertebrates, etc. The idea is that these opsin genes diversified prior to the diversification of vertebrates. So if we have gene duplications that took place prior to the last common ancestor, Luca, then Luca would have multiple forms of the gene. The trees for these two genes should be the same. So what you do is you make an unrooted tree and you root to where they connect. So if you have, let's look here. We have gene A1 and gene A2. Gene A1 and A2 split prior to the lineages that contain those genes. That is groups 1, 2, and 3. So we could say, let's say bacteria, archaea, and eukaryotes. So if we do an unrooted phylogeny of gene A1 and gene A2, all in the same phylogenetic analysis, you could see the lineages will be repeated twice in the phylogeny. That is, group 3 has a gene copy of A1 and has a gene copy of A2. So then what you do is you just root the phylogeny where the unbranched phylogeny, essentially where they connect. So here we'd be rooting the phylogeny where the star is. So pull down on the rubber band string at that point. And then we have the rooted phylogeny at the bottom where we're seeing the organismal phylogeny agrees in the phylogenies of these two different gene copies. And that is how we have insight into rooting the tree of life that Luca represents the most recent common ancestor of bacteria, archaea, and eukaryotes. Now, not all genes support the same rooted tree. And this is a little bit of an issue in phylogenetics. And if we go through and look at these different loci and infer a phylogeny from each of these loci individually, we could call these gene trees. What we see is that archaea, eukaryotes, and bacteria, most of the genes support a tree where archaea and archaea and eukaryotes share common ancestry, but we have support among these genes for these alternative trees. What's up with that? What's driving that? Well, lateral transfer is common in bacteria and archaea. So you think about the way of genetic exchange in bacteria and archaea through conjugation, and this can confuse matters. So here we're looking at a phylogeny that's called the organismal tree. And you could see that if there was genetic exchange between species C and D, we would then infer the wrong phylogeny. So the lateral gene transfer will make the recipient lineage D appear to be most closely related to the donor lineage C relative to its most recent common ancestor that it shares with B, all right? And one way around this is to think about phylogenies where you're looking at multiple genes and seeing what the consensus among the multiple genes are relative to the individual gene tree that's giving you a tree that disagrees with the consensus phylogeny. Now here's a great example of lateral gene transfer from archaea to bacteria. So archaea and eukaryotes are thought to share a common ancestor. And looking at this, the gene at this locus here the phylogeny inferred from the DNA sequences. And we can see the one blue clade of bacteria is nested well within the green archaea clade. So we would argue this is not the actual phylogeny, but this is a phylogeny that is reflecting a lateral transfer event of the archaeal genes into those bacterial lineage. Hence, at that locus, it appears that these bacteria are nested within the clade of archaea. Now, it's thought that the early evolution of life was not tree-like. There were all sorts of what we would call these reticulations. So often, the early part of the tree of life is referred to as the web of life. And there's a really interesting popular book that came out, a book for the lay audience that came out a couple of years ago, David Quammen's The Tangled Tree. If thinking about the early evolution of life, if you find that interesting, and also the story of the folks who made these discoveries is really well laid out in this book. Okay, now I would argue that one of the most important discoveries made in organismal biology ever was the phylogeny of the major clades of life carried out by Professor Carl Woese, the University of Illinois, Champaign-Urbana. Now, Carl Woese's work, really kind of his first phylogenetic work came out in the mid to late 1970s. And it was literally, it just blew people's minds. In that, his phylogeny argued that essentially prokaryotes are paraphyletic, whereas the archaea, the unculturable bacteria, the unculturable prokaryotes, were the sister lineage to the eukaryotes. And bacteria were sister to that clade. And the most recent common ancestor for all living organisms, so this is Luca that we're pointing to here, dates to more than 3.5 billion years ago. And the common ancestor of archaea and eukaryotes dates to more than 2 billion years ago. The fact that prokaryotes were shown to be paraphyletic was fundamentally altering in the way we not only think about the tree of life, but think about mechanisms that have generated biodiversity that we see today. Now, you could think, yeah, we know there are 3 major clades in the tree of life and kind of wrap it up and put it away. But just a study that was published just 4 years ago. In fact, the first year that I was teaching BIOL 104, this paper came out. And this paper has blown many minds. So this paper is based on metagenomics, which basically you go out and take environmental samples and essentially build and sequence genomic libraries from just environmental samples, so soil, water, etc. And then it becomes a bioinformatic process of piecing together all of these genomes that are sequenced. Now, these are not organisms that are cultured. These are bacteria and archaea that are not cultured, but their genomes are being sequenced from these environmental samples. Now, in this study, they assembled about 30,000 genomes. And they were able to reconstruct 1,000 genomes from the environmental samples. And the phylogeny that they inferred is not from the genomic sequences. It's inferred from the ribosomal protein gene sequences, not ribosomal RNA, which is often used to infer phylogenies among these major groups in the tree of life. And the red dots basically mark lineages that have never been isolated or cultured. Now let me take you through the architecture of this phylogeny. So here are the eukaryotes, okay? Here are the archaea. And as we're going to learn in our next lecture, archaea are paraphyletic relative to eukaryotes. So there are archaea that are more closely related to eukaryotes than they are to other archaea. And here is what we know of as the bacteria. Now, what's the one really incredible result from this study was a whole major group called the candidate phyloradiation that was discovered in this study in 2016. So here we have eukaryotes, here we have archaea, here we have bacteria. What is this? What is this group? Your guess is as good as mine at this point. What I'm trying to show here is that there are still fundamental discoveries to be made with regard to the composition of even the major groups in the tree of life on Earth. So what is descendant from Luca is still an area of active discovery. Okay, so in closing lecture, and I thought this rather timely, given what we're experiencing with the COVID outbreak, is let's talk a little bit about viruses. I usually just dog on viruses in this class, in that I just say they're too weird, they're probably not alive, and leave it at that. But I just put together a few talking points to think about viruses. So they may or may not be considered true organisms because they do not have a physiology. They depend on other life forms to reproduce. And it's clear that viruses have evolved multiple times. Okay, so the origin of viruses is something that's happened multiple times. And viruses are likely escaped components of cellular life that have evolved a parasitic living. And viruses exhibit a diversity of genetic structures. Now, viruses are abundant and they're vital to many ecosystems. There are up to 10 million viruses in a milliliter. That should be milliliter, not millimeter, of ocean water. And every day, 1/2 of bacteria in the ocean are killed by viruses. So most viruses are attacking bacteria. And the daily explosive population cycles of these bacteria are what drive ocean ecosystems. So viruses are very critical to ecosystems on the planet. Okay, so what should you know from this lecture? Basically, the general timing events, and I have the wrong slide here, so what you will do is the proper slide is at the beginning of the lecture. So I will make sure it is at the end of this lecture for the key points that you should be thinking about for this lecture. So until next time, Bio 104 students, be safe and be well.